Okay, let me pull that up. And you'll save everything and then send it to all the participants?
Yes.
Okay, thank you.
All right, everyone, it's about 11:27. We're just going to give a few more minutes here for remaining participants to join us before we get started.
Sounds good.
Hello, everyone. Thank you for joining us today for Successful Practices Discussion in Biology. We're going to get started in about 3 minutes. Hello, everyone. Thank you for joining us today. We're going to get started in approximately 1 minute. Hello, everyone. It's about 11:31 a.m., so it's time for us to get started. Welcome and thank you for joining us today for the Successful Practices Discussion in Biology. I have a few housekeeping items to discuss before we begin the session, and I would also like to go over the agenda real quick before we get started. So during the webinar, unless you're one of the presenters, I would recommend that you keep your video off until we get to an open discussion section to preserve bandwidth for those that may have some issues with slower bandwidth. During the presentations today, if you have questions, there will be time at the end of each presentation to ask questions, but then also the chat box is available for you to type questions in as the presentations go forward. So the first, for our webinar today, Dr. John Plenifish is going to be facilitating the webinar. And the first presentation of the day is a joint presentation from Bonnie Futrell and Dr. Sandy Specht from Sinclair Community College, and they're going to be talking to us about their remote gel electrophoresis experiment. The second presentation of the day is from Brent Stauffer at the University of Cincinnati, and he's going to be talking about HyFlex solutions for fall of 2020. After the two presentations, we'll just have some time for an open discussion where we can talk about any items that participants may have questions on, or things that they're experiencing as they're moving into the fall semester. And so with that, I'd like to turn this meeting over to our facilitator, Dr. John Planifish.
Thank you. I think that we're going to look at a couple of specific examples of remote learning and how that's going to be either is being implemented over the summer. So Bonnie Futrell and Sandy Specht are going to talk a little bit about something that they've actually been doing over the summer in terms of remote learning, and then preparing for fall from Brent Stauffer, and then we'll throw it open to a discussion so that you can bring forward things that you're doing at your institutions, and also ask questions about what kind of things are happening at other institutions in the fall. And this is really to help us prepare for the fall, because I have a feeling that we're going to have students who are going to be needing to do their lab work remotely, and that's going to be a challenge. However, I think there are solutions, and with that, I'm going to turn this over to Bonnie Futrell and Sandy Specht. Thank you.
Thanks. Yeah, John, you're right. There are a lot of exciting things going on, and Sinclair Biology has been doing a lot of those, been trying to prepare a lot of those. And so today I'm going to talk briefly about some of the experiments that we're working on for the fall, and some of specifically the gel electrophoresis. That's going to be our main focus. So my name's Bonnie Futrell. I am the biology lab technician, lab manager at Sinclair Community College.
My name is Sandy Specht, and I'm an associate professor in the biology department at Sinclair Community College. I'm also an assistant dean of the science, mathematics, and engineering division. I just wanted to tell those of you in attendance who aren't familiar with Sinclair, we're located in our main campus is in Dayton, Ohio, and Sinclair offers more than 250 associate's, bachelor's degrees, and certificate programs. And we have 3 strategic priorities: alignment, growth, and equity. And our motto is find the need and endeavor to meet it. So at Sinclair, we really try and meet the needs of the students. And, you know, that includes keeping an eye on keeping tuition costs down for books and learning materials, and keeping our labs affordable to the students.
So our department has 16 full-time faculty, 30 part-time faculty, and 4 full-time staff. That includes 3 lab techs. And Becky Jarvis and Kelsey Zelsky the 3 of us have been working really hard to try to work with faculty to develop these remote labs. We have, when we're on campus, we have 12 teaching labs, and our department offers 17 individual lab courses. So when this COVID thing hit, we kind of made a quick transition into, you know, setting up our dining room tables with lab experiments. So we've been working really hard with these. And so we're pretty excited about the information we're going to share. Here are a list of what we're going to be doing in the fall. Our ecology, we only teach that in the fall. So these are all experiments. Dr. Danette Richards has been the main person setting these up, deciding what was going to be taught. A lot of our ecology is using a lot of stuff that the students have at home. So this is at a pretty minimal cost. We are supplying some of the scientific equipment, but again, at very minimal cost to us. Dr. Richards also set up all the Majors experiments, and those involved a little more cost for us, some of which people don't have to do. I think are the most expensive thing on that list we sent the students columns for our column chromatography experiments, which is it's a really cool experiment, but I'm not going to go into that. our main focus is to talk about the gel electrophoresis, which Dr. Speck has been involved with teaching, and I did a lot of the background for that. So that's why the two of us are both sharing that information. So the electrophoresis.
So I'm going to talk to you a little bit about the gel electrophoresis. Many of you who teach biology are familiar with this technique and understand how important it is to have the students complete this. The learning objective for this lab was to show students how to run and interpret DNA fingerprinting gels and help them understand manipulating DNA and loading electrophoresis gels and all the applications that you can use for the electrophoresis technique. So we wanted to translate this into something that they could do at home safely. So as you know, these labs are not only helping the students understand how to manipulate DNA and understand more about the chemical properties of DNA, but also RNA and proteins. So Bonnie's going to talk to you a little bit about the experimental design.
So when we were trying to decide what we were going to be able to send home to the students, one of our coworkers, faculty members, Susan Lucan, had some experience doing this experiment at home. And so she suggested we give this a try. And so immediately I went out and found a vendor that offered this kit to purchase. And I purchased it for $66, which obviously is not cost effective for us to send $66 kits to the students. When I received the kit, I immediately saw that all the components could be purchased a lot cheaper. We're always looking for bargains, trying ways to save money. And also we wanted something that if we were sending it to students that were maybe a few states away or military or whatever, we wanted things that we didn't really need to get back. So the first time we did this, we actually we put the combs from the manufacturer in these kits and asked for the students to send them back. And the combs are about $20 each when you do this. So we were trying to figure out how to do this. So ultimately we got the supply cost down to $9 again versus the $66 from the vendor. We did end up giving our students batteries for this because we were worried about if they had any kind of economic hardship with the COVID. We wanted to make sure that they weren't struggling to get supplies that they needed. So our ultimate cost was $15 a student, but again, that was $6 per student for the batteries alone. So we've worked that out and we're still working it out. I mean, it's still a work in progress. So here's the supply list. We obviously had to develop and send them the protocol, electrophoresis chamber, all this stuff. We have an image of all the items. So I don't know how well everybody can see this. So one of the things that we thought was pretty cool was we bought this soapbox, and again, this was this was developed like a hybrid of what the scientific supplier gave us and what we found online. And there are a million DIY online electrophoresis experiments that you can do. So we kind of took the best of both worlds and combined those. And one of the things you see on the soapbox, which I thought was pretty clever, is we actually melted little notches in the soapbox so that the students are putting their comb exactly where they need to put their comb. And you can also see that we put a negative and a positive on the box so they know how to hook up the electrodes. And you can't really see it, but we actually did put little notches in there so that when they put their electrodes in there, they're positioned properly too. So we really tried to make this as user-friendly as possible. The one thing you're seeing in this photo is I'm working with one of our engineers on campus, and they are actually printing 3D combs for us. And I think he said that material cost is maybe 15 cents. So we're excited about working with the other departments and getting the cost down and not requiring students to send these items back. So the protocol, so after doing all this research, figuring out how to run the experiment, running it with the supplies from the vendor. Like I said, turning my dining room table into a lab, one huge lab experiment. I had to develop a protocol, and I wanted to include lots of photos for the students. I wanted to, again, make this as user-friendly as possible. So the students, we actually just send them paper clips. That's what they use as electrodes. So you can see I've included photos of how they need to unfold the paper clip to make it work. The photograph here is showing a picture of again, the vendor comb, which we're not going to continue to use, but hopefully replace that with these 3D printed combs. And then also, one of the things we thought might be tricky is the assemblage of the batteries. And so that has a step-by-step photos of how they need to put those together. And we did include a lot of safety instructions. Again, this is a work in progress too. So we're updating the information on this, and we'll be updating some of the photos on this also.
So what did the students actually do? Well, they took those M&Ms. This was very, very cool. And again, I feel like I really want to give Bonnie and the other lab technicians a lot of credit, because they spent a lot of time finding all of the correct equipment and at the lowest cost, and figuring out what worked, which helped us as faculty a great deal, because we didn't have the time to do this because we were in the middle of teaching. But what the students were actually doing was melting the dye color coding off of the M&Ms, and the students had 6 different wayboats, and we gave them an aliquot of, or we gave them a handful of M&Ms, 3 of each different color, and the students just had to use a running buffer to melt the candy coating, the color dye off, and then they used transfer pipettes to actually load the samples onto the gel onto the electrophoresis gel. Here it is in the soapbox, and you have all set up and ready to go. And the students had to connect the electrodes to the batteries, and then what they were actually doing was separating those dyes in an electric field to separate the dyes based on their chemical properties. And then they had to make observations and answer questions, homework questions, and they had a quiz on this experiment. So one of the most important parts, I think, of the assignment was to have the students, take pictures, or one picture or multiple pictures of their results, and upload them with their homework assignment, so that we could first see that they completed it. And second, we could also have a chance to discuss their results with them. And just like we would in lab, well, why didn't things work out the way you expected them to? And let's talk about, ion concentration and current and different things. So we had a chance to talk to them about their results. And then it also gave us the added benefit of seeing actually what the result was. And we gave these pictures back to, I gave these pictures back to Bonnie, so that then she could meet with the other lab techs, and they could talk about troubleshooting the experimental protocol so that we could make improvements when we use this experiment in the fall. So this was one of our best students, and she had a just picture perfect result. And so you can see how all the different colored dyes separated out, much like the DNA, different pieces of DNA, different size pieces of DNA might separate. But as with everything, sometimes there are fails. So Bonnie, would you like to talk about the less than
perfect pictures? So from a lab tech perspective, and my role in this whole process, we're trying to, again, make this user friendly. And so when I was working with Dr. Speck on this project, I told her, or asked her, to please get the failures and a lot of the feedback from the students, because this is exactly what I need to know, so that I can troubleshoot so that it doesn't happen again. So one of the things that we didn't have control of in this was the type of water that the students used, because we didn't think it was, we didn't think we should be sending water, plus it would add to the weight if we were shipping items and so forth. So in the protocol, I did suggest that they use distilled water. And if that wasn't an option, to use some kind of filtered water, either RO or even a Brita filter, whatever, to remove some of the metals and solids in the water that they were using. So with the image on the left, we're not quite sure what happened. This was a student who we actually sent her a second or supplies for a second gel setup, and she kept saying that that it would not solidify. So I'm not sure if it was the water. The image, it would have been helpful to have a couple more images, so we can't tell if maybe she didn't put enough buffer in it or what happened. So this one I'm working on trying to reproduce. It may be tricky and I may never do it. But then the image on the right, we think the student had some issues with loading the gel. It almost looks like they maybe punctured the bottom of the well when they loaded. So we're not quite sure, but we as lab techs, we love doing this stuff. And so not that we want the students to not succeed, but we want to know when they don't so that we can fix it.
So after the students had completed the experiment, because I am a science enthusiast, I wanted to kind of collect some data. And so Bonnie and I worked together to put together a survey to give to the students, and just to kind of get some feedback from them to find out what types of water. So I talked to Bonnie about what kind of information would be helpful, and that was one of the things she said, what type of water did they use? I also asked them things like, what type of surface did you run your experiment on, and what were there any difficult parts of the procedure where you had stumbling points. So I kind of just put together a couple little graphs to show the results that we got from the student feedback survey. So you can see that there was a mixture of well water, tap water, distilled water. So even though we asked the students to use distilled water, they didn't have it necessarily available, and may not have had the means, or may not have been even going to grocery stores in the middle of the pandemic. So they just used what they had on hand. And we still had students who had successful outcomes with the Brita filtered water and the tap water. Question number 3, I'd asked the students about whether there were any parts in the procedure that were difficult to follow. And for the most part, really the majority of them said that, no, it was fairly easy to follow. And that was because Bonnie, in developing that lab protocol, had pictures and had instructions so they could see exactly how to put the batteries together and so on. So for those students, I also had an open-ended student response question just to get a little bit more feedback. And we had a lot of really positive feedback about students having their parents help out with the experiment, and they were teaching their parents about what they were doing. So that was really exciting to see. Lots of students who said they really enjoyed doing it. And the one, I think, benefit to having the students each doing their own gel, as opposed to what they would do in a face-to-face lab, is that instead of having a group of 4 people running one lab running one gel, a group of 4 students loading a gel and running it and looking at the results, each student was responsible for making their own gel. They got to do all of the different parts and load all of the gel samples, and so it really put all the responsibility on that student. We did get some students giving us feedback that it would be really nice if they had access to the teacher when they were actually running the experiment. So that's something that we're going to kind of take a look at. If this is something we might want to do when we're live streaming labs in the fall, run these gels synchronously for those students who want to have the faculty member, online with them. They can be setting things up and walking away from their camera and getting water and doing things, and then we can kind of talk to them about any of the points that they get stuck on. So the last question that we asked was, would you have preferred to have more at-home labs, more online lab simulations, or was it about the right amount? So for this class, which was a second-year genetics class, the students we found a lot of online lab animations, so things like HHMI and lab exchange where the students were actually kind of clicking and dragging to simulate a gel, setting up a reaction, restriction enzyme digest or something. And then we also had the at-home kits. And we had about 40% of the students saying that they would love to have more at-home kits, and about 40% said it was about the right amount. Some things like bioinformatics, that lab translated very well online, and about 20% of the students who wanted more or all online lab simulations. So this was kind of nice feedback for us, because we kind of went into this just doing our best, and it looked like our best turned out pretty well. So thank you very much for your time. Here is our contact information if you want to reach out to us directly. I know myself and Bonnie are happy to help with any of this if this is something you want to adapt. So feel free to reach out to us. And thank you very much for your time.
Thank you.
Thank you. Are there any questions for Bonnie and Sandy? I'm going to start by asking one real quick question. For your course, so for your course, the genetics course, how many kits were there versus online, so other kinds of online solutions? Was this the only kit, or were there others?
This was the only kit that we actually did, that the students actually did at home. Okay. All the rest were online, right?
Can I ask also for the intro bio courses, how many kits were there? Were there kits, or were there other solutions that you said in one case there was stuff at home they had access to?
Bonnie can speak to that a little bit more than I can, because she works with the professor, but for the majors level, the biology course, that's what she was talking about. Our general education biology class, we have developed an online lab, so we have virtual microscopes and things that we use for our general education biology. But for the majors biology, Bonnie, do you want to?
Yeah, what we did with that, because and then minimal scientific supplies, and so we put together like a little lunch bag full of things that included pH dipsticks and the column for the column chromatography. Trying to think off the top of my head some of the other things that they did, but so we just put everything together in a little baggie, and we did ask the students that could pick them up on campus to save us with shipping costs, and then we shipped the ones that they couldn't pick up. So we sent them like transfer pipettes and things that were useful to them that maybe they wouldn't necessarily have at home. So
right, so maybe 5 or 6 for the majors biology. Labs were done at home, and then the rest were online, sounds like. But we feel like that was a pretty successful, we had to really scramble and move quickly, and we were sending out kits, in week 3 or 4, so, or having the students come and pick them up at one of our buildings in our drive-through, have them drive through and throw the kit in the back of the car as they sped through.
Okay, so there's a couple questions. One of them is just a technical question. The question was, do you have us muted? Is chat the only way to ask a question? And I asked if somebody could unmute themselves, but they don't seem to be able to do that. Should they put their hand up? Be unmuted? Zoe?
That's a...
I just unmuted Linda. Linda, are you able to speak?
No.
Yeah, we can hear you.
Oh, okay. It still shows my microphone is muted.
Yeah, there's a line that stays through the microphone, but it sort of turns orange. I know I did that 3 or 4 times too.
Oh, okay. I'm used to
orange
using
when
Zoom.
you...
I'm used to Zoom.
Yes.
It says my video isn't on, or is it?
Yeah, your video's on. We can see you.
Okay, I'm sorry. I was just frustrated with the platform. Very, very interesting.
It's a learning experience for all of us, we're finding. Get to experience what the students experience.
So
I was a few minutes late because I had trouble getting in. So are you doing online labs this fall?
No, we're doing a combination of I'm sorry.
I mean, are you doing all virtual this fall, or are you doing face-to-face?
We do have some face-to-face labs.
Okay.
There was a question from Paul asking, do you think sending supplies to the mail will be an issue if they're sent through UPS with their current issues?
There were no issues with the supplies we sent. One thing we were concerned with, for the ones that had the more expensive equipment, like with the columns, we were concerned if we put those like in a bubble mailer, that somebody in the mailroom or at the post office would they would smash it. So what we ultimately ended up doing was we bought these, I think they're called mug boxes. So like a 4x4 box, and put the supplies in it. There it is. But again, most of the students pick their supplies up. So that wasn't an issue, but the stuff we were mailing, I mean, we sent a buffer that is completely safe, and we had all the supplies. Well, the reagents were pre-aliquoted, so we sent them exactly how much buffer they needed. We sent them exactly how much agar they needed, and again, the agar is a pure grade agar. I mean, they could eat it if they wanted to. I wouldn't recommend it, but it's completely safe. And so that's one thing that we've struggled with when we think about. Like for the chromatography, for instance, the paper chromatography, can we really legally send them acetone? We don't know. So we just said, okay, the students need to provide their own.
There's some request also in the chat. Would you mind sharing some of the activities you listed for the ecology course, perhaps via email? Are these also online simulations or kits?
These will be kits. Our list, the list that we gave in the presentation, those are all going to be part of a kit.
So maybe sharing some of that through to us, and then we can send it to the group if you'd like to. That would be
Yes.
helpful.
Yeah.
Absolutely. Or anybody's welcome to email us directly too.
Okay. One last question, I think, and we'll go to the next presenter. But one last question: did you include a mix of a color as an unknown? So
the brown was a mixture. The orange M&M was a mixture. But no, we didn't. We just basically had them just melt the colors that they had on the M&Ms, but in the fall, we're thinking about different dyes we can use, and potentially, part of why getting feedback from the students was so helpful, and having Bonnie run these, experiments is so helpful. She can test different candies, so we're, different items, so we can make some different gel electrophoresis experiments for the students to be able to use, so they have a different experience.
We have been working on some recipes to actually just send them very small amounts of these different dyes. And in that case, John, you're right. We could actually, because they're not looking at the M&M it came off of. So we could actually set up one that was an unknown and kind of like a whodunit DNA simulation kind of thing, only with colors. And then obviously they wouldn't need any kind of a UV light box, or they wouldn't have to stain the gel to see anything. It would just be the colors from the dyes.
So I'm just going to circle back before we go on to our next speaker. The question about the mail was about the delivery issues, current delivery issues. Did you have problems with delivery issues?
No.
Because right now they are running a little bit late on delivery, USPS.
No, and we gave the students who live out of state, because we had some students who were quite a distance from campus, so they couldn't drive through. We mailed them labels that they can just ship their products back. So we're hoping that we'll get most of these kits back, just like the soap boxes and things that we can reuse.
All right. Well, thank you very much. If there's additional questions, we can bring them out in the open discussion portion of today's meeting. Next, we have Dr. Brent Stouffer from the University of Cincinnati, who's going to talk about HyFlex solutions for fall 2020.
All right. Hi, everyone. It's nice to be here, and great to hear from Bonnie and Sandy about their creative solutions for at-home gel electrophoresis. We're going to shift gears a little bit and kind of go at the kind of the course-wide level, which I think will complement each other quite nicely. And my hope here is that, by sharing some of the thought processes I've gone through as I develop the course and think about creative solutions, that we can all just kind of pick and choose what might be appropriate for our respective courses. I think there's some strong power in being able to share ideas and pick and choose different elements and incorporate that into our course room design. Particular, as you can see, my ultimate conclusion for this upcoming fall is that my course will be doing what's called a HyFlex solution. And so if we going through this, we're all kind of deciding, or a lot of you probably have already made a decision as to what your class is going to look like for the fall. But, we got our 100% online, whether you can do it synchronous or asynchronous. And then kind of within an in-person course, you can split your section into 2 sections in different rooms, or you can split into kind of 2 groups in a hybrid sort of way. And I discovered kind of earlier this summer, early on, kind of this HyFlex solution, and kind of intrigued me and kind of led me down a path and a thought process. And so that's really what I just want to share with you all. And I'm going to hit on a few different things, though, because as we think about our design for these different in-person labs, there's a variety of different processes to think through. And so I'm going to introduce very quickly HyFlex to talk about my specific HyFlex model, think about how we can kind of sometimes reorganize our curriculum within our lab courses to best accommodate the unpredictable fall semester ahead. And then also I'm going to touch on some classroom setup and movement patterns as well. So that's what I'm going to be hitting on today. But before I do so, starting off just very broadly, perhaps a lot of you have heard of the HyFlex model at this point, but if you haven't, it actually just means kind of a combination of a hybrid flexible component, right? So very broadly, kind of a hybrid component where there's some in-person options, but also some online. And where it gets its name for the flexible part is that traditionally a lot of students can sometimes choose whether to attend online or whether to attend in person. And so therefore that's where we get kind of the name HyFlex. It's actually been around for a while. I was actually surprised when I kind of looked into it, as more and more programs across the states and the globe are trying to build up their online presence. This kind of HyFlex model has been provided as a solution, maybe not to need to have 2 separate courses, but instead have a single course which has more flexibility within that particular course. Now, there are some variants of the HyFlex model, and one where you literally let the students choose whether to do it online or whether to do it in person. The way I'm kind of going about doing mine ultimately is this second point, where it's a little bit prescribed by the instructor. In other words, for this upcoming fall semester, all the students in my course will be coming in person at some point in time and will be attending online at some point in time as well. But the students get to decide kind of within their group which week they're going to do the in-person and which week they're going to do the online portion. So mine's a little bit more prescribed. On a side note, we are offering a couple sections that are 100% online for my particular course for some of our international students and students with health concerns. But for the students that do want to come in person and get some of the hands-on skills, this gives them a little bit of flexibility, but still hopefully provides a kind of a safe manner for the students to be able to do so. Now, before I get into some of the nuts and bolts of the plan itself, I wanted to give you all a little bit of context into the type of course we're talking about here at the University of Cincinnati in our biology department. So this is kind of an introductory biology lab, Biology 1 lab. Very high enrollment, right? We have almost over 950 students or so every fall semester come in and out of this class. Biology majors, but also plenty of other colleges within the university are also take this course. We normally have to operate this course alone in 4 different rooms across 11 different time slots. So we have a little bit more of the kind of cleaning concerns of the large physical space as well, and also just the sheer number of students that go through the hallways throughout the week and throughout the semester as well to think about and consider. Personnel-wise, we have me, full-time staff member, and then this particular fall, I think we're going to be able to have like 17 TAs who are graduate students and 2 adjuncts to pull off the 40 lab sections that we're going to be offering. So one of the things that means for me, for example, is like it's not only a decision I have to make for myself, but I have to be able to make sure that I can train all of the individuals involved how to do this properly and in a safe manner as well. We meet ultimately 13 times throughout the semester, which encompasses a total of basically 6 different teaching modules that they learn in Biology 1081L. And as we sit here today, just like everyone, we had that kind of spontaneous adjustment in the spring semester. So we've had about 4 weeks of this course that was taught kind of in an asynchronous model where the students could kind of do the assignments whenever it was convenient for them. And then this summer, I've been teaching sections that were 100% online in a synchronous model where in some of the cases, in some of the labs, I actually taught from the room myself, but they were supposed to come to a particular time slot. So, now inventing the wheel a third time, essentially, what I decided for the fall semester was this HyFlex model. And so one of the reasons why it's HyFlex is one of the first things I did is I looked at each week by week and decided ultimately that 5 out of the 13 weeks, we are perfectly fine without pretty much any curriculum changes being 100% online. I think in this kind of introductory biology lab section, one of the biggest things that we all teach our students is how to think like a scientist, right? How to design experiments, how to work together as a group of scientists and training these scientists. So, 5 out of the 13 weeks, we can move rather seamlessly to 100% online environment. So I made a decision, well, if we can do that, let's just be online for those 5 weeks without any problems. And so that leaves 8 remaining weeks where the students will be coming in person in some sort of shape or form. Now, I want to point out that, as you can see, normally our classroom capacity is right around that 24 to 27 students range. At the University of Cincinnati, we have kind of a design and planning committee who's reduced the capacity to make sure we can socially distance ourselves within the rooms. And so the capacity's technically been set to 11. Now, even though it's been set to 11, I made a kind of a personal decision because of the sheer scale at which we operate, that we can get away with having 6 students in the room instead of 11. And so basically what we're going to do is 8 out of the 13 weeks are going to be this kind of HyFlex model. And as you can see on the slide, within each group of 4, one of those students are going to be in person, and the other 3 are going to be simultaneously attending virtually online, basically being able to interact via the webcams from the 6 different students and the instructor within the classroom. And so that's kind of their eyes into the lab setting, and therefore they have some hands that are able to actually conduct the experiment as well. So that's the overall model. And what I think is really was helpful for me is to think about the different perspectives of the different personnel involved. So this means for a student, they are attending online for 11 of the weeks, right? They can see the instructor via WebEx, Zoom, Teams, whatever your favorite web conferencing program and platform is. They can still have their group-specific conversations in the breakout rooms. They can still have a lot of good conversation about experimental design and complete a lot of their in-class assignments in these breakout rooms. But in terms of in-person, this means that an individual is really only attending in person 2 to 3 times throughout the semester, and that's it. And what this means essentially is that I made a couple decisions. Is like, hey, while they're there, regardless of what lab they are conducting, the first time that they show up, they will be trained on all sorts of different hands-on activities and devices and pieces of equipment, especially our microscopes, right? That's one of the biggest things that they learn in our first semester of lab is how to use the microscopes. That's one of the most difficult things I think we've all encountered that is difficult to simulate in a virtual world. And so hopefully we can get them all at least in once, at the very least, to get trained on all of this. And then hopefully we can get them in a second time as well to actually test them on what we've trained them to learn. So that kind of builds in some accountability as well for being able to learn some of what they're learning. All right, now from the group's experience, so now we're talking about the group of 4 within the classroom. That means that at the very least, there should always be one person on behalf of the group that is in attendance. This also means within the group, what I'm going to try out this particular semester is to have more prescribed roles within the group so that we can make sure that all the group members are staying busy. In other words, that 100% of the work isn't just done by the one student that's in class, right? That's a big concern that we have in this model. So we want to make sure that we prescribe some roles, right? You can get creative, I suppose, with whatever names you want. We have an executioner, which sounds a little bit more dangerous than it is, but right, the person who actually conducts the experiment and executes the experiment, a scribe who can write down the notes and instructions from the instructor, stopwatcher who kind of time management, and then kind of a manager who oversees some of the executions of the project as a whole, et cetera, et cetera. So hopefully this keeps everyone engaged in a more lively way than perhaps if we didn't create some of this infrastructure. Now, from the TA and adjunct instructors experience, so remember, we have some annual adjuncts, for example, who are teaching 6 to 7 sections of this course just by themselves. The TAs typically teach 2 sections. And so my hope is that by doing this HyFlex model, instead of having like a separate online course and a separate in-person course or something to that effect, that hopefully this means just kind of having one prep, right? You can prepare just once for teaching both simultaneously online and in person simultaneously, and hopefully have a little bit more flexibility based on whatever it is that COVID throws our direction this upcoming fall. This hopefully helps students to stay engaged as well, kind of allowing them to interact with the experiment and do the experiment. I really like, Bonnie and Sandy's plan where the students are actually doing something on their end of things. I found for myself this summer, there were multiple times where I ended up kind of having to do the whole providing them some dummy data. And, that there's something a little bit less exciting about doing that. So hopefully this HyFlex solution also reduces the need for that as well. And then the coordinator's experience, my own experience, is also allows me to still have like a single large merged Canvas course, not necessarily a separate course for the online sections, a separate one for the in-person sections, is kind of a single one. And then again, something that I just have to think about is like how to train all the TAs how to do this. And so hopefully having one training session is going to help us reduce some of the workload and the burden there as well. I want to talk briefly about the breakdown of the curriculum and just kind of some of the thought processes. I know some of the names for these modules won't mean a lot for everyone, of course, but kind of helps you kind of see some of my thought process and some of the thought processes you might use and utilize. So first off, like I mentioned before, 5 out of the 13 weeks are 100% online. And then notice one of the things that I did is the very first week I decided to make 100% online. All right, so that allows us to hopefully iron out some of the wrinkles ahead of time, get everyone adjusted to the technology, and also use that opportunity to get everyone assigned to their appropriate weeks that they're coming in person or not. I also actually switched the order of what the modules that we normally have to try to create put our module that is the most amenable to moving 100% online if we need to towards the end of the summer when there's kind of a little bit more concern about the flu kind of interacting with COVID and potentially having to adjust to going online again. So I think anytime we kind of can rearrange some of the order of our curriculum to make it amenable, I think that's super helpful for all of us as well. And then notice from the in-person end of things, this is where I have kind of the first, each of the group members getting trained on different weeks, and then each of the group members getting tested on those different weeks as well. So we'll see what COVID throws our direction, but hopefully everyone can at least come in once to at least get trained on those in-person skills, but hopefully, again, get tested on some of them as well. Here's a list of items that I kind of just decided to kind of just throw onto the PowerPoint slide. Again, a lot of this was just for me to be able to process this and think through a lot of this. So I actually created this PowerPoint a month or two ago, if nothing else, just for me to be able to think through every nook and cranny of this. So we have 2 different stations, one in which is going to be a whole bunch of items at the student's table that because they're smaller items, right? These stations are going to be basically kept there throughout the semester, or at least the basic, essentially. So we need to have like small items at each table so the students don't have to move around a lot. And then some larger items like the power supply for gel electrophoresis, the rig itself, a thermal cycler for PCR, just so students can at the very least see what they are, learn how to identify them, and at least talk about how they would be used, would be at kind of an alternative station where they can be a little bit more spread out. So that's kind of the list of items that I have in mind for that. Then I want to briefly, this is going to, I'm going to go through this part pretty fast, but again, this won't be the room setup that all of you have, of course. I'm, of course, fully aware of that, but hopefully it helps everyone think about if you are overseeing some labs and if you are having some sort of in-person component, how to think about designing the classroom and setting up the labs in different ways. So this is our typical setup that we have, the instructor at the front, 6 different tables where there are groups set in. Instead of the normal 24 students, we of course have 6 different students. I got just 1 at each person, but I actually created this flow diagram to show students like how they need to walk into the classroom, right? What do their movement patterns ideally need to be, and where will they actually be seated once they actually do arrive into the classroom. They'll be walking in staggered fashion so that they aren't all kind of crossing paths at once. And then our hope is that in the ideal situation, and the staff member and I have been talking through a lot of this already, we think for the huge majority of the labs, we can get as much equipment as possible on each of those tables to make sure that the students don't necessarily need to move around to all the side benches like they normally would have to do so. If they do need to do so, we started to map out as well, like where they would move to and from. We're going to have these stations clearly labeled. The students will know exactly how to move from where they're seated to the station. If there are some scenarios where we only have 3 stations, we've got that kind of figured out, and they can kind of take turns going to the station so that they can maintain their social distancing and of course be masked up and potentially have some face shields if they want to. And then, this is kind of a quick reminder, like you also think about redesigning the class or the curriculum itself. So normally we have students do the CELSAP experiment and a burning experiment where they determine the percent organic of the matter that they are given. In this context, so that they don't have to move around the room as much, basically half of the groups are going to do the CELSAP experiment. The other half of them are going to do the burning experiment. And that way, we can still talk about it, have some good discussion about it, but not necessarily need to have them cross paths nearly as often. So that's a big thing to consider. If we ever do need a station, a single station, we don't think this will happen very often at all, but they would just have to take turns or consider having the TA pass it out instead, since the TA will be masked up and have a face shield on as well. And also, I'm even mapping out for the TAs like where some of the safe places are to stand and still be socially distanced from the 6 students that are in there. When they're at the stations, where should the TA stand, and where can the TA kind of move and still sort of see what's going on, but still be able to maintain some safe social distancing as a result. Some of you might consider some plexiglass options as well. And remember, plexiglass is not only to, yes, it might protect a little bit of the virus itself and the droplets and such. Really, it's more of a behavioral thing, right? If you see plexiglass, you're going to know to stay in your lane. So we're considering that for some of our ability to kind of separate the stations as well, to make sure that students know exactly where to go and where they shouldn't be going as well. And potentially at the tables themselves, if you feel like that's appropriate. But I think really the biggest thing here in this particular slide is we're going to make sure the TA, him or herself, has a shield at their desk so that students know they can't just walk up to the TA anytime they want to, which is very contrary to what we, of course, normally want to do. But hopefully that helps protect the TA instructor or the adjunct as well. So, there's a lot that I just kind of threw out there, but I hopefully something kind of clicked with what you might be doing or what you might be useful for your particular course. I think in general, what I've learned the most is, right, we all got to be flexible. But my hope is that that the HyFlex option might be particularly flexible. One thing that I think we all know is that, it's easier to make changes now as opposed to kind of midway through the semester. So definitely process your plans with other people outside of your own course. Obviously, everyone here is doing that to some extent already, which is super powerful and useful. And then, my goal too, and especially for these large enrollment courses, like this PowerPoint isn't just for me. This PowerPoint isn't just for the sake of talking with all of you right now. This PowerPoint also allows me to kind of show my students that like what they're going to be doing when they come in to hopefully help them remember to stay safe and to protect each other, and also for TA training purposes as well. So if there's anything about this PowerPoint that might be useful, even if you want to just use the diagrams just to give you all a starting point, like by all means, like reach out for me via email. You're welcome. I'm happy to send the entire slides over to you if it helps you not have to create all the diagrams and stuff or anything like that. I do have a baby girl arriving in 5 days, so I might be a little tied up and a little busy in 5 days from now, but you're happy to reach out if you have any questions. And yeah, happy to take any questions right now.
All right, thank you, Brent. Very nice presentation, very helpful thinking about the HyFlex model itself. I do have one question that I would like to just ask really quickly before I get the other questions. You talked about webcams. So where are the webcams located? There is an issue right now, as you're probably aware of buying new webcams. So where are your webcams located in the lab? And could you expand on that?
Yeah, that's a great question, John. I think if you read a lot of some of the information online about the HyFlex model, is it very strongly like talks about how, having a webcam up in the corner of the room so that everyone can see the entire class, or if you have some classrooms or lecture halls that are already equipped with the camera. I am honestly expecting the huge majority of our students to already have webcams and mics ready to go. At the University of Cincinnati, I think that's kind of been an expectation that students are going to have. And I know that a lot of people are working really hard to provide students with webcams and microphones if they don't have them already. So there are some plans for that. But we really hope to provide, if absolutely needed, one webcam and one mic for each room for the TAs to have. So yes, sorry, Sarah, in your chat, webcams that are just on their laptop or tablet. Yes.
Right.
Built in, so that of course there's some quality control issues there, but my hope is that it gives them just enough insight as to what's going on in the lab that and they'll be hearing from the instructor as well. Let's not forget that the instructor will be on camera and be able to talk through some of the different things that are taking place as well.
Thank you. Question was, do you have a document cameras?
Yeah, we do have a dock camera in each of the labs, and then we have a projector in each of the labs. So I've thought about too, kind of projecting the WebEx room itself so that students can see on a very large screen, like everyone attending virtually. I think that would be fun in some regards for the students to kind of feel more of a community feeling. And then we also could use the dock cam and have that shared as well. I haven't thought about, I don't know about the quality of kind of rotating the dock cam to see the class, but it sounds like that would maybe something to test out. So that's a great idea.
Sorry.
Go ahead.
I've used the document camera in that aspect many times in my anatomy and physiology classes. And it does work, but it matters on the quality of the webcam and whether it actually fits out to your projector. But it's a nice way to be able to show and experiment to students as they work through it in those individual setups. And our biotechnology entire program, we've shifted all of our labs to the front end of the semester so that we can get the techniques out of the way, because it's pretty likely that things are going to be difficult near the end of the semester when you want all those techniques, right? So we have to teach them those statements and the techniques all at the beginning. So that's a very challenging part too. Sorry.
Yeah, I think I saw, I could be wrong. I think I saw recently Miami University is actually staying online the first couple weeks. So
That is correct.
yeah, so that they're kind of, it's kind of a double-edged on both ends kind of difficulty.
One question I put in the chat and someone else had was, what if the student assigned to show up that day doesn't come, or if you have a student who just doesn't want to be there in person?
Yeah, so the reality is with a course with 950 plus students, that's going to happen a lot. You know, there's that reality of that happening. And so I hope for every single week, the execution of this is going to be like via sign-in sheet that they're creating and planning in week 1. And then for every single week that is this kind of high flex, partly in-person component, that there's a backup person ready to go when possible. The other reality is, like, worst case scenario, if there are groups 1 through 6 and group 6 ends up not having anyone in person, I can just have the group 6 join the breakout group for group 5, and they just have a larger group than normal for that particular week. So
Okay.
I'm hoping that can happen as well. And that's what makes it more flexible, I think, in some regards, is because that is going to happen at some point. And so having some flexibility built in, I think, is certainly helpful.
Okay. Yeah, if you know in advance, you can get a substitute, but if they just don't make it that day.
Yeah, and that's when we're going to have to just spontaneously move them into group 5 virtually instead. So
Okay.
There's a question from Justin. Will you have a makeup lab option?
Yeah, during the normal semester, we because we have 11 different time slots. We normally allow students, if they reach out to me or one of the TAs, to, like, if they can't attend their normal Tuesday morning time slot, they can attend the Friday afternoon one instead if it's excused. And so we basically plan on still doing that if they need to. And then if they can't make it at all, yeah, of course, absolutely. One of the biggest things that we've been emphasizing at UC within our faculty senate is to not punish students for lack of attendance, right? Like, grade them on participation, not necessarily attendance. So we're going to be extremely flexible. And the reality is, if a student were to reach out to me and something changes halfway through the semester, and if they decide that they don't want to come in person at all, I'm going to personally work with that student and make sure that we have a backup plan to get them to maybe do some extra video quizzes and watch some extra videos to try to kind of look at some of these videos, more, asynchronously than if they were in person.
All right. Oh, okay. So, thank you, Brent. This has been very informative. I would like to now move to throwing this open to a more open discussion. And just to help start that off, one of the questions that I think is on everyone's mind is, how are you going to be assessing your students throughout the courses? And I can see there's a list of discussion topics, but I actually want to start with the one that's down on that list of topics a little bit, which is a question about how are you assessing your students throughout your courses, given you're either having a lot of online part of that they're doing. How is this being done? And there's also this question about, as you can note in the chat, there's also a comment about altering how I grade the attendance portion. You could feel free to unmute as well and talk.
I'm still trying to decide how to do some of these things. So some of your questions I'd be curious to see the answers to. I normally do a class where I lecture part of the time and then we do some type of activity. So I think if I'm doing half face-to-face and half online, maybe I could do the lecture part during the face-to-face and have them submit the activities for points in which case they wouldn't be punished if they weren't there. But then I'm thinking, or would it be better to do the activities while they are present and do the lectures online? So I'd be interested in seeing how people are approaching that.
I've done all my lectures as asynchronous, so they can access them whenever. All of my testing is synchronous, and that's for two reasons. Canvas has problems sometimes, and so I can go in and support them. And if questions don't load or anything else, I can make sure that works. So I have them all with a specific test time, and I always have Dropbox activities or completion materials that they have to do. The best example is my 2752, my AMP2 course, which isn't a Gen Bio course, but I have them like cut out paper models of antibodies and antigens and things like that. And it's a lot of crafty stuff that then they can take a picture of, but it's stuff they have around their house to show that they have done it. Blood vessels especially, that schematic of blood vessels was always the best way to learn it. So I walk them through in a set of videos on how to draw it, and then they turn in those drawings to me via Dropbox. But there are a lot of different pieces, and so I'll have like a few moving parts labs, and then a few look at this anatomy, whether it's 3D anatomy, et cetera, labs within each lab grouping, because they only have so much time.
Okay.
And in Gen Bio, we also are doing some more than I would probably like, but more simulation labs, because we don't have a lab tech. We don't have any like support system. It's just me trying to build labs for 3 or 4 different courses along with lecturing. So I do a lot of simulations from like SimBio, and so those do have questions and turn-in pieces, and then there are scientific articles that they need to read and analyze. So that also shows me completion. So those are the lab assessments that I have, and I still do have some lab practical exams from that online format that I do.
Okay, so you are totally online?
We are totally online for some of our courses, and we are hybrid for others. And part of that mattered whether or not we could get the plexiglass in place in time, and whether or not scheduling worked the way it was supposed to. That's the whole different smile.
Yeah. I only have one course at the moment that I think I'm going to be meeting half and half.
Okay.
So I'm kind of curious as to how to approach that half and half.
Well, as I said in chat, with our half and half, we took, and that's all our biotechnology courses have to be half and half, because there are very specific techniques that our employers need for these students. So we front loaded all the techniques, which doesn't fit as well with, what you're lecturing, but all the techniques are front loaded, and then the hypotheticals, theoreticals are more at the end. I'm sorry, I'm at North Central State College.
Okay. Yeah, I agree with you for the lab there. I was wondering how people are handling lectures if they're half and half.
Oh.
And fitting that in with the assessment. Because I usually took, in the past, I took attendance, and whatever activity we did that day, I graded.
So there are
half lecture, half activity. So I'm wondering how to translate that into a hybrid situation where half are there one day and half are there the other.
Well, you can have them all do the same activities, just in different ways. If you're doing, whether it's a creation or drawing activity, or but the worksheets, where they're working through to build out, and it just has slipped my mind because I'm trying to say it.
I guess what I'm saying is, if I'm just meeting half the people without anyone online that same day, would I be better off lecturing and then have them do the activities as on to be turned in later, or would I be better off having the lectures online and have them do the activities when they're closed?
So the classic flipped classroom discussion is that the activities should be done in person so you can support them and guide them through. And so they have to prep the material ahead of time and know where they're going. What you're talking about sounds more like a flipped classroom in my eyes.
So I want to just say at our institution, University of Toledo, if you have a class that is split like that, where there's half the people there for the lecture, you're also supposed to stream it at the same time to the group that's not there. Now,
Okay.
whether your
That
activity,
was a whole nother possibility, which I need to discuss with the administration.
so that's how it's being handled here. Of course, the question of if there's activities you're doing in the classroom, that's a little different, but that have to be then turned in. That's different. But certainly for the classes where half the students are there one time and half the students are there another, and it's a lecture class, you're supposed to stream the lecture at the same time.
Okay, so I'm not sure if we can have the technology to do that at this current point, at least for all of our classes.
Yep.
So we were talking possibly about a flip-flop of one versus the other, but for safety reasons, I might not want to do the in-class activities in class. As I said, I'm unsure at the moment.
So I'm just be curious to hear from the group how you are actually handling or planning for your labs this fall. What does your current plan look like? I mean, we've heard in some detail from what Cincinnati is doing for their particular intro labs. We've heard again, from Sinclair College to some extent what's going on there. But I'd like to hear a little more broadly what's happening on other campuses. We also heard from NC State, at least in terms of what's going on there. So feel free to.
So at Cuyahoga Community College, I don't teach. I am the associate dean of the department. We are going to offer the general biology and a face-to-face, and I really love Brent's model. I just email you and say send me your slides. So I'm actually going to meet with Joe Koch, who teaches that course. We are going to have some face-to-face. He's going to have two labs that are face-to-face and one lab that is totally remote. And so I love your model of videotaping and sharing that with the students that are in the remote environment. And I don't know exactly how he's going to execute this, but I love your model. I don't think he's on this presentation, but I definitely will share your slides with him with your permission. I like the model. We did the same thing. We did social distancing. We walked in with rulers and measuring tapes and made sure we knew where students were going to stand. I haven't had that walkthrough with Joe just yet. He wasn't part of that. I was just working with the compliance department. And we are going to probably develop a model very similar to what you have shown.
Anyone else?
We're down to 12. We're down to 12 students in our space that allows, that's why we have multiple labs for that one instructor, because only 12 students would be allowed based on what compliance has told us for safe distancing. We possibly would be using shields, but definitely masks will be required, and we'll work out the logistics later on this month. Next month we'll work out the logistics of how students will rotate to the lab. Again, probably using your model as a template. So thank you.
When are you opening? When do you start?
August 24th.
Okay.
So we have a little time to because I have not been back to the building more than 3 times since March 18th. It's a science building with about 14 labs in the building: chemistry, biology, physics, earth science in the building, and so it's largely a science lab building. Very few classrooms spaces. The classroom is in the rest of the campus.
So, anyone else?
At Stark State, it's depends upon the professor. I think we're all doing slightly different things. I've chosen to do synchronous lectures and then do split the lab in half. I have them do stuff in lab in person. The other half is doing, I found a good book from Prince George's Community College in Virginia that's designed for at-home stuff. So like week 1, I'll be doing something in person. the other half of the class will be doing something out of the book that I have. And then the next week we'll flip flop. It's kind of how I've chosen to do mine. I've also got a purely web version too. So that's asynchronous. So again, it depends so much on the different professors and how they've chosen to do it. But I said, I like synchronous, and it works for me. But the labs, we're just going to flip flop between in person and at home, and then the next week it's the reverse.
What is the at-home resource that you have?
It's called Hands-On Biology Laboratories for Distance Learning.
Can you put the title in the chat so we can have it?
It is a private published. I found the book initially on Amazon and end up finding one of the authors, and she gave me the address of the private publisher.
So you have required your students to buy the book?
Yes, it's not bad. It's about I think it's about $35.
Okay.
And as a title, it's by giving you a there's the authors. The Moore Crawford was the lady that I got a hold of, and it's done by this company here. And that's the publisher right there. So if you need something, I can figure out how to get you the contact of the publisher. If you want that, send me a message, and I can figure that out. I have it somewhere in my email. It was originally published through Freeman. Freeman dropped it, so they picked it up and started publishing it on themselves. So there's a couple really good labs in there, and those are the ones I'm going to use in there. There's a real good one on macromolecules. There's a good evolution one in there. There's, again, multiple really, really good ones in there, and those are the ones I'm going to choose to use for the alternate weeks for my students.
I taught biology too for freshmen in the spring. I don't the fall class, but I did the sow bug behavior as an at-home lab, and it actually worked out really well. I usually have the students design their own experiment, and they were actually very creative. I did have them take pictures of their setups, but they came up with all kinds of containers and setups to look at preferences and the little roly-poly things. And I didn't require anything more than they might have in their kitchen.
And we ended up sending a small lab kit home with them. Some stuff that to finish up those labs. It was about $15 for the lab kit. And then I had them do videos of them doing short videos of the labs as a way to have some idea that they actually did that lab.
So I'm going to ask just a quick question. It says, what did you learn through your summer offerings about delivering, it says geology, you should say biology, lectures and laboratory courses. So for those of you who are doing laboratory courses during the summer, which I assume in most cases were primarily online, although maybe not, what did you learn that was useful? Now I know Sinclair's already talked a lot about what they learned, but I'd like to hear from other people as well.
I didn't teach this summer.
Did we have anyone that was?
Yeah, I did the Gen Bio this summer, and the people who survived did fairly well, but my dropout rate, I started with 29 people. I ended up with 15 plus 1 incomplete.
Was that fully online this summer, or was it?
Fully online.
Online?
I was using that book that I mentioned there, so. Yeah. I said there was a large withdrawal rate.
I had the opposite. Hopefully you can hear me. I taught anatomy and physiology too for Edison State Community College, and I did not have a very bad dropout rate. And I found that most of the assignments, including some of the online labs, had to be asynchronous for summer. since there's so much information being done in the summertime versus the fall, which is spread out in those 16 weeks.
Yeah, my loss rate wasn't much higher this summer than it was last summer. So, and this was technically done asynchronous, but I had about half my class showing up at the same time every day, and I kept showing up at the same time every day. So it was kind of both synchronous and asynchronous.
Do you find that students, and this is a more general question for people that have been doing either lab or lecture, have you been finding students prefer the asynchronous or the synchronous delivery?
I think it's easier to slough off the asynchronous. Oh, I'll listen to it at some time, or I'll do it sometime. The synchronous, I think it's, it comes closer to mimicking a face-to-face. Again, that was my preference, but I'm not saying I'm right, but different things work for different people. But like I said, synchronous was my preference.
In the spring, I had a lot of people put off the asynchronous and then really did a poor job on what they did turn in at the last minute, versus a few who finished in the first few weeks and did excellent jobs.
I just found it interesting for my asynchronous class this summer. I told them I'll be online 10 to 12, Monday, Wednesday, Friday. I did the lecture and recorded on Collaborate and Blackboard, then published the recording. But I had about half my class showing up at the same time so they could hear it live and ask questions live. And then they'd go back and listen to the recordings.
Okay. That's good to know.
So I had the one girl, she did very well. She's like, yeah, I think she came to all except one lecture. She's like, yeah, I'll listen to your lecture, your PowerPoint, your questions, and then I'll go back and listen to the recording later.
I did learn in the spring that you have to be a lot more specific in your instructions than you do in the classroom, where you can clarify things.
Yes. Found that out the hard way a few times.
Yeah. And sometimes it was just a small detail, but it made a big difference in interpretation.
Especially if you use the breakout rooms, because once they're in the breakout rooms, you can't see what they're doing all the time. So it's impeccably important to really give clear instructions in the main room, and then visit them as soon as possible in the breakout rooms, just to make sure that they're starting off on the right track if you are particularly using breakout sessions at all.
Yeah, I like the idea of the breakout sections. The asynchronous was where I ran into the problems of either not getting it done or not understanding directions.
Sandy, we can't hear you. Still can't hear you.
Can you hear me now?
Yes.
Sorry, I have another switch that I sometimes bump. I feel like I'm stating the obvious, but it does seem like the live stream teaching and trying to translate all of this material to an online format is a lot more work for the faculty, and it takes a lot of energy, and you're sitting at your computer a lot longer and having to meet with students via Zoom and also learn so many technologies, different interfacing, software programs to meet with students, Zoom and Teams and different, WebEx. And I feel like a lot of credit should go to all of those faculty members who have done that, because it is, and I don't know exactly why, but just sitting in front of your computer and talking is harder. And the other thing I'm kind of struggling with is, can we make it mandatory for students to have their cameras on? Because I found that for some classes that I teach, all the students have their cameras on, and I like that. It's easier for me to see their reactions and see that they're actually there, but then for some classes, all the cameras are off. And I'm kind of wondering what people are doing in terms of
Yeah.
classes or their plans are for fall for.
I have to agree with
I don't
Kirsty's
know.
comment. It's a bandwidth issue sometimes when all the cameras are on. So I've had meetings where we've had to turn our cameras off simply because if they're on, everything slows to a crawl.
Yeah, we had the one professor who lives in a more rural place, and he had difficulties even having a meeting with 2 or 3 of us on, with the camera on. So I like the idea of having the cameras on, but I also know that, I've got 5 people in this house doing exactly the same thing I'm doing. So we run out of bandwidth pretty quickly.
Sure. That's good feedback. Thank you.
I'll throw out that this summer, teaching 3 different lab sections and then also participating in a bridge program, which was more of a lecture kind of format. So with the caveat that I was teaching for class sizes of about 20 to 30, I actually did make it a requirement for students to have their cameras on unless they emailed me otherwise. And so that way, if they have issues, by all means, they can keep their camera off. They can, we can occasionally like have cameras on only in the breakout rooms where there's only 4 to 5 students instead. Because if I told them, if you give them any permission at all to not have their cameras on, by the 2nd or 3rd meeting, all of their cameras are off. I mean, all of them. Even if their bandwidth is perfectly fine, they keep their cameras off. So it's, just something to be aware of. And I asked them after the fact, at the end of the semester, if they appreciated me forcing them essentially to have their cameras on. And they actually really appreciated it. They really appreciated it because for so many of these students, like this is the most number of people that they are seeing on a daily basis is in a classroom setting. So just, I'm just throwing out there another perspective. Obviously, if a student has bandwidth issues or doesn't have a camera, let's of course accommodate them as much as we possibly can. But I think the students do miss being able to see their classmates.
I mean, some of it's a question of how much difference in engagement is there if you see everyone else in the class as a person, or if you see them as just little circles with initials or whatever, right? I mean, so there's that question. But I do have concerns about issues like bandwidth in particular, and not every student is going to be using a computer. Some have, as somebody mentioned already, only have a cell phone as their access. And
Yeah, about a quarter, I do stuff in downtown Canton, and probably of my 2 classes that had 40 students in, I probably had 7 or 8 at least that there only was either a hotspot or a cell phone, the hotspot provided by the district or their own cell phone. They didn't have a computer, so that makes life interesting.
I was asking about etiquette in the chat. Does anyone have like a list of things that they ask students to do, like try to be in a quiet area without, their, dog barking, and whether you allow eating, which I find distracting.
Linda, I probably had 4 different students this summer who joined the class while they were on a trip to vacation, like they were in the car attending class. So it's not a bad idea if you do want to have them in a, focused environment to consider having some etiquette, general rules of thumb in your syllabus.
Yeah, because I've even been to some meetings with faculty where you have a grandchild running around and crawling over them, and it gets very distracting.
My wife had one listening to her lecture driving back home. He lives on the West Coast, so he's actually listening to her lectures live on the phone as he was driving.
Right. So we're getting towards 1 o'clock here. There's a few more minutes. If there is any other comments from the group that they want to share?
I was just going to throw out there that my colleague, Dr. Duslos, pointed out a book called The Core Rules of Netiquette, and it was by Virginia Shea. So that may be something to take a look at. She kind of, went over it. She went over that portion with the students because we team taught our genetics class this summer. So I'm less familiar with the content of the book, but that's something you might look at. Get some ideas for how to present that to students.
Okay. And it is a good point that perfect etiquette could be a state of substantial privilege. Yes.
Justin,
One thing that I found that was really incredibly useful this summer. I had my students pick up their lab materials because of how ordering went. It wasn't until about halfway through the summer semester, but it gave me an individual check-in with each of the students. When they drove in to pick up their lab materials, I could talk to them directly and say, hey, what's going on with this? Or have you been able to schedule it these? And I also had that in a start here quiz that I asked them, what times are you working? Are you an essential worker? How do we work around these particular difficulties? Because you got to have the money to pay for the course. So being able to check in and talk to them one-to-one for just even 2 minutes, it's like that office hours visit, only it's effectively a quote-unquote forced office hours visit where they have to talk to you. So taking those just 2 extra minutes, keeping them in line and talking to them, I thought it was gold.
As we're wrapping up too, I want to, for those of you new to WebEx, if you don't know, you can go to File, Save, and then Chat, and you can actually save the chat box if you have any desire to do so, in case there's any resources that were shared that you want to hold on to.
And I'll also say that at the end of the webinar, we'll also send out a copy of the WebEx from today to everyone that was in attendance.
thank you. Well, then we'll include the chat as well, the whole thing. Michelle?
Zoe, does it include the chat?
The streaming link that I will send, you'll be able to scroll through the chat. However, at some point, the streaming link I send probably won't be available once it's off of WebEx, but we'll still have the video on our website as a YouTube video.
All right.
So for a couple weeks, you'll be able to view the chat, but we're saving them on our end as well in case.
Okay, so people can request the chat if they want from you.
Yes.
Okay, thank you. All right. Well, thank you for all attending. I think it's been very helpful. I've heard some very useful information. I think today, and it's gonna help us, at least here in our institution, going forwards and making this a better semester for our students this fall.
Great. Thank you, everyone, for attending today, and have a great rest of your day.
You too. Thank you.
Thank you.